new area to consider called stereoisomers. Right? They're a little bit different to positional and chain isomers, right? In that they may come off the same carbon, but they have different properties, right? Because there's different arrangements in space. Because remember, we're talking about 3 dimensions, not 2. Yeah? So stereo refers to the 3-dimensional shape, giving isomers different properties. 2 different types of stereoisomers. One is called optical, one's called geometric. And let's go through them. Starting with optical isomers. These are also referred to as enantiomers. Say that again. Optical isomers or enantiomers. So when I have a carbon center with 4 different constituent particles attached to it, I have a chiral center. When I have 4 different things attached to a carbon center, I will have 2 possible non-superimposable mirror images. Is that clear? No. Okay. Right. What does the word superimpose mean? Putting on top of one another. Right? So superimposable image would be something like me taking this key and having a replica of that key, and I'd be able to slot exactly one over the top of the other. Yeah? Make sense? So a clear example of non-superimposable mirror images are your hands, right? So if I take my middle finger, line it up with my middle finger, only my middle fingers line up. Yeah? Make sense? That's not superimposing. If I do this, you're not superimposing the objects. That is taking the mirror image. It's not superimposing. Take the mirror image to superimpose means to take one of the objects, align it with the other. So my middle fingers are superimposed. My other fingers are not. Yeah? So 4 different particles, or 4 different constituents attached to a carbon center is called a chiral center. Chiral coming from the word hand. So a chiral center is a carbon with 4 different things attached. So think about, well, many parts of your body. They are mirror images, but they do not superimpose one another. Your left foot, your left shoe doesn't fit your right foot, for example. They're called optical isomers because, right, they polarize when I apply light through a molecule, they will polarize light, they will change the direction of light in different ways. Vivi's not working. Oh, how annoying is that? Right, let's pause. Okay, so hopefully you can see this a little bit more clearly. So I've got a carbon center, yeah? Carbon center, 4 different attachments, right? Green, white, red, yellow, yeah? So they're mirror images, right? So I've got the yellows pointing together, the whites are pointing towards me, the reds are pointing towards the camera, and the green is up above, yeah? So let's see if I can superimpose these. I'm going to line up the green, I'm going to line up the yellow. So the green and the yellow can superimpose, but what do we notice is the red and the white are not superimposed. Yeah? So we call that a chiral center. Now, if I have 2 of the same things, so I'm going to replace the yellow with green. So in this case, I've got 2 of the same. Now, I'm going to rotate these around. I can rotate them a little bit further, and there we go. I've got perfect superimposed images. So when two of the same constituent particles attach to the carbon, it means that this is non-optical. So it's what's called an achiral center, a non-chiral center, if two of the things are the same. If I go back to the yellow, there is no way in which I can rotate this such that the molecules become superimposable. So I can align the green and the yellow, but the red and the white won't be. If I go to try and align the yellow and the red, right, then, oh, these are the exact same molecule, yeah? So if I swap two of those positions, right, then there's no way I can rotate this such that I get the same molecule. So I'll rotate that around. Let's line up, say, the green and the white. Line those together. Again, non-superimposable. So they're called optical isomers because if we put it through a polarizing plane, they will result in polarizing the light in different ways. One is called the R or the plus, one is called the S or the minus enantiomer. Now, you're not required to name it. Yeah? Not required to label the plus minus, not required to label the R or S forms. You're only required to recognize chiral centers. Chiral objects, left and right feet, these shells. There are achiral things, such as a chair, perfectly symmetrical. If I fold that, I would superimpose the two. Butterfly, achiral. Yeah, left hand is exactly the same as the right hand. So we've got chiral versus achiral. Remember, the prefix a means not chiral. Shoes, scissors, etc., so on. There we go, I showed you that example. 4 different attachments, non-superimposable. Okay, so there's the example. You will be asked to identify a chiral center. So let's have a look here. So if 2 of the attachments are the same, it's achiral. All right, let's take a look at this example. Identify any chiral centers in the following molecule. We must have, thank you, 4 different attachments. So if we look at this carbon, 1, 2, 3, all the same, achirals. Looking at this, I've got a hydrogen, I've got an OH, I've got a CH3, and I've got this whole big bulky group. 4 different attachments. This, therefore, is a chiral center. Here. Yeah? Here, 2 hydrogens are the same, therefore achiral. Here, it's not a chiral center because I've got 2 bonds associated with that oxygen. Yep. So can't have 4 things. Yep. It could have 3, but can't have 4 because I've got the double bond taken up. So not a chiral center. So the only chiral center here is this one here. Right? Let's look at the following molecule. Let's go through them all. Is this chiral or achiral, this carbon here? Achiral. Why is it achiral? Right, I've got an OH group coming off both the top and bottom. This one here, achiral, 2 hydrogens attached. Yeah, this one here? That is chiral. Hydrogen, methyl group, ethyl group, bulky group. Yeah, this one? Achiral, achiral, achiral. Right, physically, yeah, optical isomers or enantiomers have the same physical properties. Yeah, they're going to stack in the same way. They are going to bond in the same way. However, they react differently. More so, they react differently if in combination with biological systems. Yeah? human bodies contain many chiral molecules. So molecules that have chiral have different effects in the body. Pharmaceutical drugs, many exist as enantiomers. Only one of the enantiomer will have that beneficial effect on the body. The other may have no effect or may have a disastrous effect. Take example, smell. These are two chiral molecules. There, designated by that asterisk, is the chiral center of this molecule, and we've represented the mirror images of these two molecules. Notice the dotted line represents going into the page. The bold line represents coming out of the page. So if you want to visualize it that way. These two molecules interact with your olfactory receptors. Yeah. This molecule here, right, will smell like spearmint. This molecule here will smell like caraway seeds, right? Some sort of, spice or whatever. So, same physical properties, different way in which it reacts with biological systems, and that's why we talk about stereoselectivity in biological molecules. Stereoselectivity. So you have a unique receptor that will bind to this, yeah? You'll have another unique receptor that will bind to this. Yeah. What is about polarized light? Plain polarized light. Where was it? Interact with plane. Plane just means within the plane. Polarized. So if we go back to this molecule here, plain polarized light just means within one direction. So, for example, when you wear polarized glasses, yeah, you have a polarized filter. You've got light coming in that's within all directions of where the waveform is. The polarized light only takes one of the planes or one direction of that waveform, and then each of the enantiomers will bend that waveform in one direction or another. Yeah? Don't really need to know that. But that's why it's called optical, because optics refer to things to do with light. All right, so stereoselectivity. Think about a handshake. Yeah? Go to shake my hand. Go to shake, keep your hand out, go to shake my hand. A bit weird, isn't it? Right? Stereoselectivity. I can only shake your hand if you also put out your right hand. If you put out your left hand, yeah, it doesn't join, doesn't bind. The receptor, right, when we think about that, the active site, when we get to talk about enzymes, is different. That's our key and lock. Those of you who've done biology. Substrate, yeah. The shape of the active site of the enzyme only binds with a particular shaped substrate. Stereoselective. It's relating to this. All right. And let's talk about a particular drug example. This is a drug called thalidomide. Have you heard of it? Yeah. Probably heard of it in great detail. One of them was considered cures morning sickness. The other, right, stereoselectivity, binds to different receptors within the body, causes significant birth defects. Why? Because that drug, right, stops rapidly growing cells. Still used today, particularly in cancer treatment. If you are pregnant while taking thalidomide, provided it's if you have a mixture of both, it's called a race mix.